Hey everybody, welcome back to NeurosciQ, the best place on YouTube to increase your neuroscience IQ. We're back at it again today with another COVID-19 video. Today we will be looking at two new papers that were published in the journal called Science that mentioned that COVID-19 seems to be binding to another receptor on our cells called Neuropilin 1. So, before we dive into that, if you're new to the channel, make sure you subscribe right now and turn on notifications so you can be notified for all our future videos. And without further ado, sit tight, stay tuned, let's roll the intro. Alright, welcome back. We're talking about COVID-19 today, and we're going to be looking at its interaction with a receptor called Neuropilin 1. So first of all, we have to understand how this science came about and why this research even started in the first place. Well, if you've watched some of our other videos, or maybe you've heard it in the news, COVID-19 is actually similar to SARS in terms of genetics. So a lot of the genome is similar in both the viruses. Now, for those of you that don't know what genetics are or what a genome is, basically the genome is like this instruction booklet that our body reads to build proteins. And proteins make us who we are. So, for instance, there's instructions in my body that tell my cells how to make my hair and to make my hair brown. And everything about you is encoded in your genome. So viruses have a genome too. In fact, viruses are just capsules of genetics. Now the thing is, viruses can't read the genome and they can't build proteins on their own. So what happens is a virus will interact with our cells, somehow get inside the cell and hijack our cellular machinery so that we start making viral proteins. So, what's happening with COVID-19? We also talked about how COVID binds to the ACE2 receptor, just like the SARS virus. But scientists started looking into the differences between the genome. So it could be a couple pages in the instruction booklet that are different, but that could lead to a huge change. Just like blue eyes compared to brown eyes. Now in this case, they found a difference in the genome that actually led to a sequence in the protein, which was a marker for a furin protein. A furin protein is like an enzyme that comes and cuts the other protein. So COVID-19 has a sequence in it that gets our body's enzymes to cut it. Now what happens when that protein is cut? All proteins have a C terminus and an N terminus, and this C terminus becomes exposed when you cut the protein. Now, after that happens, the sequence that was exposed actually is the same sequence that binds to these receptors on our cells called Neuropilin 1 receptors or Nrp receptors. So this is important, and the important thing is that the sequence actually was on the spike proteins on the COVID virus. So if you look at the COVID virus, we have a circle. And then if you look at all the pictures online and all the animations, there's these little prongs coming off of it, and those are called the spike proteins. Now those spike proteins are exposed to the exterior environment. And what happens is they have the sequence that calls the enzyme over to snip it, and once it's snipped, a sequence is exposed, and that sequence will bind to Nrp receptors on our cells. But could this happen, and does it happen? Scientists, when they found the sequence and they realized that that was a thing, decided to explore whether or not it's actually binding to the Nrp receptors in our cells, and if this plays a role into the way COVID-19 enters our cells and takes over our cellular machinery. So, let's talk about what happens. Again, the steps are, we have this COVID-19 capsule, and it has that sequence that gets cut by furin. Furin cuts the sequence, and then the sequence actually has to go through another step, which was proven to happen in another paper published in Cell, and that is it gets primed. with TMP-RSS2. So we prime it after it's been cut, that way it can't go back together again. And then that sequence, that motif that binds to our NRP receptors is exposed. This is called the C-end rule because it's the C-terminus of the protein that is exposed and that will bind to NRP-1. Now, we know how that happens, but what about NRP-1? What is NRP-1 and where is this receptor in our body? Well, this is a receptor that's important for cell signaling, and it's a transmembrane receptor. So what that means is that we have our cellular membrane, so if you have a cell, you have an inside of the cell and an outside of the cell. Part of the receptor, if it's transmembrane, will be inside the cell and part of it will be outside. And what this does is it allows signals to go from the inside to the outside or the outside to the inside. That way the environment can communicate with the cell. So this is what happens when things bind to the receptor from the outside, which is what actually happens with COVID that can bind to the NRP receptor. So the NRP receptor plays roles in our bodies for signaling for angiogenesis, which is the vascularization. So it's when blood vessels form in our body to get blood to different places of the body. It also is important for neuronal growth. It's important for axon guidance. So our nerve cells, our neurons, have these things called axons that attach to other nerves so they can communicate messages. And so axon guidance and telling an axon where it should grow is guided by NRP. And also vascular permeability. So they seem to be on cells important for circulation, but also on neurons and also cells in the nervous system. So the structure of the NRP receptor, it has two A domains, two B domains, a C domain, and then the part that's in between the membrane, and then a part that's inside the cell. Now the part that actually gets binded by COVID-19 is the B domain. So keep that in mind for some of the research we're going to talk about. The first paper did a series of 4 experiments. In the first experiment, what they did was they took these cells, these like empty basic cells with no receptors on the surface, and they added ACE2 receptors, and then they added ACE2 receptors and NRP receptors to some of them, and then some of them just had NRP receptors. They then wanted to see what would happen with COVID-19 in comparison to this virus, another virus, VSV, that seems to enter more cells. It's a more robust virus. So what they saw was that we need ACE2 on the cell for COVID to enter. COVID wasn't just entering cells with no receptors, it wasn't entering with NRP alone, and it also wasn't entering with TMP-RSS alone, which was the transmembrane primer protein that keeps the protein from coming back together after it's been cleaved. So we need ACE2 to enter the cell. Next, they saw that if you put ACE2 and TSS2 in the same cell that they created, you increased the entry because now the protein was able to be primed. And this was just kind of a reproduction of the results that were seen in the other paper that was published in Cell. What they also saw with this was that if you added NRP to the cell as well, you also increased the entry to the cell. Then they wanted to look at cells that already naturally have ACE2. So they used these cells called KAKO cells. And what they saw was that if you add NRP artificially to the KAKO cells, we would increase the rate of infection of the cells as well. So overall, the first few results have been showing that, yes, the interaction with ACE2 that we saw in the past is actually happening. Yes, the TSS2 interaction is valuable and it's needed. But if we add NRP to the cell, we're also increasing the entrance. So it seems that there's some sort of three-way interaction that's bringing COVID-19 into the cell. It can happen without the NRP, but the NRP is enhancing the entrance to the cell. So what they did next was they wanted to make sure they proved that the interaction that they thought was happening was happening. So how do we do this? Well, what they did was they made antibodies for the B domains of the Nrp receptor. So the Nrp receptor, we said the B domain is the special domain that COVID needs to bind to get in. And to prove that, they made antibodies for the B domain. So our immune system, if you have a foreign invader, it will make antibodies that bind to it and block it. So in science, you can do this by using animals like rats and injecting the protein that you want, and then they will make the antibodies you need, and you harvest those antibodies and you can use them in your research. So that's exactly what they did. They created an antibody that would bind to the B domain, and then they incubated the cells with the antibodies so that the Nrp receptor would be blocked, and they wanted to see the difference of entry to the cell. So they did this actually with silver molecules that were attached to the special domain that enters the cell. So remember, the COVID-19 has the C end that enters the cell, so they stuck that C end onto a silver particle, and then they put it and wanted to see if it would enter the cell. Now, if you blocked the receptors, B domains, using the antibodies, the entrance to the cell didn't happen, and infection was reduced. So it suggested that the antibody was working by using the silver. Then they did it with the actual COVID virus, and they saw the same thing. Infection was reduced if the antibody was blocking the receptor. Another experiment actually did a very similar thing. What they did was they actually used a known antagonist for the Nrp receptor, and they saw the same thing. When the antagonist was bound to the Nrp receptor, the infection rate of COVID-19 dropped. Now, the second experiment in this paper actually looked at what would happen if we mutated the furin cleavage site. So we said we have this special site on the COVID-19 spike protein that needs to get cut, but if we mutate it, then the scissors don't know where to cut. It's like you have a dotted line that tells you to cut here, but now all of a sudden the mutation and the dotted line's gone. There's no more indication of where COVID-19 should be cut. So they mutated it, and we can see that in figure A. We see the mutations happen, and we see that there's no cleavage happening through this protein analysis because there are no S1 and S2 units. So the spike protein isn't getting cut into an S1 and an S2 anymore. So what they saw afterwards was that the COVID-19 with the mutation, it could only infect cells that had the ACE2 protein. And if there was a Nrp protein, it would not enter at all. The normal COVID-19 usually could enter with Nrp, but in this case, because it was mutated and that site didn't exist anymore, it wasn't entering the cell anymore. And then they saw that the greatest rate of infection happens if you have all three factors, so ACE2, NRP, and the TMPR. Again, they used the antibody, and we see in figure G and H that the antibody is only able to reduce the binding of the wild type. So the one that they mutated, it had no effect on it because it was already a wonky virus, but the virus, the original virus, would not bind if the antibody was present. The third experiment took silver again, and they took that peptide sequence that binds to the NRP receptor and they put little mutations in it. What they saw was that only the one with the actual sequence was taken up, which we would expect. So then they took the one with the actual sequence and they wanted to see what cells it would enter. And interestingly, it didn't only enter the olfactory epithelium, which we know COVID-19 is entering our nose through our nasal passages, but it's also entering blood vessels of the cortex. So what that means is the little vessels that supply blood to the surface of our brain are able to be impacted by COVID-19, and this leads me to question and just speculate on whether or not this might be why we see aberrant coagulation in COVID-19 and maybe why we've seen things like strokes and encephalopathy. And then finally they looked at tissues post-mortem and they saw that COVID-19 increased the receptors, the number of NrP receptors on the surface of the infected cells. So that was the first paper. And they did a lot of thorough research and they went through many different, it might have seemed redundant examples, but they're just trying to drive the point home. Now what helped was that at the same time, on the exact same day, a paper with very similar results was published. Now, it approached some of the things different, so we'll just take a quick look at it. This one only went through 3 experiments, but if we look at the first experiment, what they did was they looked at the S1 and the S2, so the spike proteins, 2 little pieces, and they labeled it with green fluorescent protein. Then they labeled NrP with cherry. So this just helps us visualize them. It's a kind of staining procedure. And then what they wanted to do was they wanted to see if they deleted the part of the spike protein that caused binding, if it would still bind. So again, we saw that that sequence, the C-end sequence, was crucial for binding because there was no precipitate, no immunoprecipitate formed if we deleted the sequence from the protein. Now, instead of using KAKO cells, which naturally have ACE2, they actually used HeLa cells, which naturally have NrP receptors. And they added ACE2 receptors, and they looked at the viral uptake, and then they made HeLa cells that were NrP knockouts, and they saw that the ones with the knockout actually had less infection. Then they did something similar with the KAKO cells, which naturally have ACE2 receptors. They did an NRP deletion, so they deleted the NrP, and they saw that there was reduced uptake, but not binding. So it seems that COVID can bind to the cell if there's ACE2 there, and it doesn't matter if there's NrP or not, but the entrance to the cell is enhanced if NrP is present. So it doesn't matter for binding, but it matters for entrance. Moving on to their second experiment, they used this experiment to show the specificity for B1 binding, and we can see that in figure B. In figure C, we can see that replacing the sequence on the S1 actually reduced binding, and then in figure D, we can see that if we altered B1, we had reduced binding. So again, we have that B1, S1 interaction that's important. And finally, they used antibodies as well. So they made an antibody for B1, B2, just like the other paper, and they made sure that it was actually binding to B1, B2 by comparing its binding to a wild-type NrP receptor compared to a mutant NrP receptor. And when they said, okay, yeah, the antibody is working, they used the antibody to see what would happen, and they saw that if you had the antibody binding to the NrP receptor, there was a reduction in infection. And then they did the same thing with an antagonist for NrP and had the same results. We block the B1 pocket, we can't have an interaction, and we can't enter the cell anymore. Now, why is this important? This is important for things like coming up with vaccines, coming up with ways to treat the virus, and just overall understanding the virus, understanding what kind of cell types it can interact with can help us understand the symptoms that might form within somebody who's infected by COVID-19. So for instance, maybe our anosmia, there were 3 predictions that we discussed in our other video, which will come up right there. But the 3 predictions of how COVID-19 is getting into our olfactory nerve and altering our sense of smell might have to do with NrP. And knowing that NrP is on neurons and neural cells could help us understand how this infection is happening. Anyhow, this might hint at many neurological symptoms that we've discussed. It might have to do with them, it might not. We don't know. But it's a new breakthrough with our understanding of the virus, and I just thought it would be interesting to share. So, thank you for watching the video. If you want to check out the 2 papers we talked about, They're down below. They're very brief and easy to read. The Cell paper that discovered the role of the TMPR is also down below. And that's all for today's video. Make sure you like, comment, and subscribe, and turn on notifications so that you can be informed when we post future videos. If you have any suggestions for future videos, also comment them down below. If you have any questions or comments about today's video, leave them down below as well. Thank you for your continued support. Have a great day, and I'll see you next time.